what Fede just mentioned is basically what I would prepare this slide actually recently. So just a little bit of the steps that I took until now, and I thought it's nice to have a picture of the place because it's a new center. We started a year ago, and it's located in Potsdam, a beautiful city. So maybe at the end of this presentation, you would be more interested to maybe come and visit us. So this is the microbiome building, and this is basically my office right here. Yeah, and so I'm heading there this new group called Microbiome Management, and it's basically just a broad term for any microbiome stuff in agriculture. All right, so usually I'd like to, when I talk about this topic, this is one of the two main topics I'm working on: plant domestication and microbiome and microbial inheritance, and I think both fit somehow nicely together. And I think usually when we talk about co-evolution or evolution, it's a bit sensitive topic when people be like, yeah, but co-evolution, how can we prove this? And I understand that, but at least it is clear that plants themselves evolved in the presence of microbes. Microbes were there before, so at least that we know for sure. And we have even evidence of like microscopic evidence. This is not Yaphilia, it's an extinct plant. And these authors here took samples and just made sections of the plant and found, maybe this is the time that I want to show. So you can see here some fungal endophytes. They identified 4 species that were present inside plants 400 million years ago. So we also know that at least they were colonizing the plants, they were inside. And we can also safely assume that just the presence inside or outside, this interaction have influenced somehow the evolution of plants. These microbes, we now know that they don't just randomly colonize the plant, they're spatially distributed. So we know that the inside of the plant or the endosphere have certain microbes, or the surface of the leaves or the roots. These are different communities, and this is just an example that we summarized about the apple microbiome or the apple tree. And you can see there's a variation in the distribution of microorganisms, both on the surface and inside of the plant. We see even variations within the fruit. So this is for some reason this was actually maybe one of the very first apple microbiome study that I did during my PhD. And at that time, we decided to look at different, maybe I should point here, at different parts of the fruit, and it was the stem end and calyx end and peel, and we even had a pulp. And we had this opinion that when we think about apple diseases, there's a lot of variations of or like certain diseases that enter from the calyx end or affect the calyx end or the stem end. And we thought, let's look at the pathology about this. And to our surprise, we actually found a big variation between these compartments within the apple fruit. I'm not showing even this year, I mean, these are multiple studies, but we did also like a sort of a global study that I will show briefly later from about 21 locations, and we still see this exact distinction between the different parts of the fruit. So that's also a good indication of that these microorganisms are not randomly present there. But despite this, so I showed some apple stuff, and some of these samples are actually here. But when you look at between species, you see it still, despite these variations, you still can distinguish between species. This is just an example that I used to make this point. It's not published yet or anything, but within each of those, there's fruits and leaves, and for grape, olive, and apple, and you still see, you can differentiate a little bit between species. And this is what we call casually the genotype effect. But when it comes even, this is the same in theory, the same, or not in theory, it is the same species. These are different cultivars, modern cultivars, so they're really relatively recently domesticated. And you still see for both fungi and bacteria, some variations that between genotypes. Again, these are fruit surface, not anything special. And this is what recently, this phenomena or this hypothesis, there's something called phylosymbiosis. And the idea here is that the composition of the microbiome can mirror the host phylogeny. So we see the distance or the variation or this similarity correspond to how far the hosts are from with their genes, their actual phylogeny. And so we see this in a lot of actually organisms, from insects to primates, and we see also, we've been interested in this for a while in plants, and we just recently also published this on wheat domestication, and we see some sort of, you can actually, this is pretty straightforward. I mean, apart from this, but it's quite obvious that there is some sort of correspondence between plant phylogeny and the microbiome. And when it comes to evolution of the plants, we also have to think about domestication, because it's sort of the same thing when we breed and select for certain plant traits. And the theory here is that, of course, this is pretty trivial. If we have wild plants and we talk about this is the genetic diversity, we select also for certain genes. And I think we all know that with domestication, you lose a lot of genetic diversity, pretty obvious. Yeah. So we have this bottleneck effect at some stage, and then from there you have some diversion, but still the bottleneck, you lost a lot of diversity. And it was hypothesized that also microbes will have the same thing. Plants, wild plants will have way more microorganisms, and then you domesticate one or two genotypes, and then from there you breed more, but still you're losing a lot of microbial diversity. But when we look at literature, yeah, some cases you see this effect. You can observe reduction of diversity from wild plants to domesticated plants. But sometimes it's actually quite the opposite. And also sometimes there's no effect whatsoever. For example, in this case, in sunflower. So what we decided in this case, we thought, let's see the effect of domestication on apple. And apple in this case, well, because I had a project in apple, so that was the straightforward reason. But we also know the background of apple. We know how it was domesticated, from where, which species contributed. And there's a lot of studies that actually quantify even the genetic contribution of different species to the domesticated apple. So in this case, I mean, this is actually an image from a collaborator that works with apple, and I loved it so much that I keep using it. And this in the middle is Malus saversii, the ancestor, the direct ancestor of our apple, or Malus domestica, which is this one. And for saversii itself, we have other progenitors, and this is where it comes a bit flimsy, how much and which species really contributed. We have a lot of studies that does quantify this, but it comes down to 4 progenitors that did contribute, but Malus saversii is the direct ancestor. And you can see here, this is another study that quantifies. You see here again, saversii comes gives rise directly to domestica, and you have orientalis and sylvestris, which is the European crab apple. And what else is also interesting is that during domestication, also domestica gave back to the wild species, because you can now control the pollination. So there was a sort of exchange during the domestication process until today. If you go to the native sites of it. So our objective for that case was obviously to look at the effect of domestication on the apple microbiome, to understand if these phylosymbiotic patterns can be observed within the Malus genus, and to understand or try to predict the origin of the apple microbe, our modern apple microbiome, in a sort of a similar way as you can do it genetically. Can we predict how much really the ancestor contributed to the modern apple? And for this, we went to the apple germplasm in the States, and there is actually the largest collection of apple. They have about 2,000 genotypes planted in a common garden, treated in the same way. It's beautiful. And of course, this is was the advantage that at least they're not grown in resampling from Asia and North America, and then you have the bias of the geographical location, which we actually observed. This is the study that I mentioned earlier. We sampled from 21 locations, and this is one genotype. It's Gala apple, and you can see that there is a big variations within due to the location itself. And so this was our design. If you look here, I don't know how to hide this part, but yeah, this is the phylogenetic tree of the Malus genus, or the species that we use. And we have here the wild species, completely not contributing to modern apple. And we had 3 species from North America and 3 species from Asia. And we had a progenitor group, and these are the species that did contribute, the Sylvestris, Prunifolia orientalis, and obviously Seversiae. And then we have Domestica coming here. And then we collected also samples from Heirloom cultivars that are still in use somehow, but and of course the modern cultivars. And you can see pictures of these species, the wild species from North America and Asia, the progenitors here, and the Heirloom or old cultivars and modern cultivars. And for this obviously was done with sequencing. So what we did, we took cuttings or shoots from the common garden and surface sterilized them in sodium hypochlorite. We peeled them, so we took only the inside, or the endophytes in this case. And then we did libraries for 16S and ITS, and were sequenced with Illumina, obviously. And we also quantified the abundance of those two groups. When it comes to results, you can see somehow, not necessarily in bacteria, unfortunately, but you see in fungi here, this is the wild group. And as we were mentioning earlier, this was contradictory to what we expected. The domesticated and progenitors had higher microbial diversity. Yeah, this is just Shannon and species richness. And you see the wild species have much lower microbial diversity. And this was also surprising when we see this. This is the microbial abundance for both fungi and bacteria. And you see that wild plants had much lower abundance or number of cells, microbial cells for fungi and bacteria. And it goes to the progenitors, and then the domesticated apple had much higher quantity. We don't know why is that, but yeah, we can discuss this a bit later. We then looked at what is so-called the core microbiome, and these are these are the 3 groups: wild, progenitor, and domesticated. And these blue ones are fungi, and the pink ones are bacteria. And the size of the circle is basically the abundance according to the qPCR. And this top group is basically shared between all of them, and you can see that the most abundant ones are shared across all the species, which is interesting that there is a conserved microbiome. But we also see that wild species had certain bacteria that were sort of lost, and in domesticated apple we have much more fungi, which probably explains this box plot that we saw earlier, why fungi, domesticated apples had more species richness, or especially fungal species richness. This, despite it, we don't see a lot of or significant difference, but it's still interesting to look at the difference between heirloom and modern cultivars in the community composition. And we see here still, I mean, maybe here it's pretty obvious. We have 2 clusters between the modern cultivars and the heirloom. I mean, maybe 1 variation, but there is a sort of distinction. However, this is not statistically significant. When it comes to the phylosymbiosis, and that was the second question we had, and so we had to build the phylogenetic tree of the species that we tested. And you can see this green group. This is basically the domesticated apple plus the progenitors. And then we have these red dots, and you can see still some outliers or some variations, but these are the wild species, this whole group, but some from Asia and some from North America. And so we built also the same sort of tree, a dendrogram with the composition of the apple microbiome. And we started with the fungal community and correlated the 2 trees together. You can see there is a significant correlation between the 2 groups, and it's still funny to see that still have the sort of cluster of the domesticated apple and its progenitors together, and then you have the wild species. Again, Sylvestris for some reason was an out group. But here for bacteria it was actually pretty nice, because you actually see a very clear distinction that is almost better than the phylogenetic tree itself. But yeah, in any case, it was a significant correlation between the microbiome and the plant phylogeny. And so we observed a philosymbiosis in the Malus group. And for the last question, we thought about this prediction, how can we really predict the origin of where the domesticated apple microbiome come from? We see philosymbiosis, we see that there is similarity, we see a shared core microbiome, but can we predict somehow how much of each of those species contributed to Malus domestica? And for this, we use this tool. I'm not sure how familiar you know these tools. This is a Bayesian algorithm that was developed to predict contamination in environments. So, for example, they use it in hospitals to know where the source of the contamination, using still this sequencing data. And we've been using it for a while now to predict the origin or the source of microorganisms. And so we did, So this is for fungi, and I hope you can still remember the colors or whatever, but this Malus cerevisiae, this is the prediction of how much of domesticated apple microbiome comes from different species. So there we did not just restrict the model to think about the progenitors only, which we know. I mean, we know that there are these 4 species, but we just gave it all the species, including the wild, completely non-contributing species. And it was funny to see for fungi that more than 50%, the model predicted that it comes from cerevisiae, which is the main ancestor. And when it comes to bacteria, we realized that we have still, cerevisiae, sure, very large proportion, actually the largest proportion, but still the other fractions were coming from the ancestors and the progenitors themselves. So there was still some sort of correspondence to the genetic contribution to domesticated apple. And I just put this again so you can remember how the tree looks like and how this contribution still happens. And in this case, for the genetics of the Malus species, we call this introgression events, that during domestication, some genes were, of course, moved from one species to another. And we made that very cautiously the word microbial introgression, because we thought, well, if that happens for genes, it could have happened also with microbes. But when we did this and we published, or we submitted this, one criticism, this is the last thing I'm going to talk about as one slide. But the reviewers just said, yeah, all this sounds really fascinating. You're heading towards one direction, that there is a meaningful contribution. And the only way to explain this is, which we also wrote in the discussion, that there is sort of microbial inheritance in it. And this was also, as I said in the beginning, it's another line that I was working on. How can you prove that? Is there any evidence of this? And so we went to do this small experiment where we went to get apple flowers from, again, the same field, closed, never opened, and we just, under the laminar flow or aseptic conditions, we cut them open like vertically like this, and took pollen sacs closed and the ovule. So this was really tiny sampling, and it was a bit difficult for sequencing, but yeah. And we also took shoot endophytes. So we have this theory, if it is inherited, there is only 3 sources: the male contribution, female contribution, which is the ovulins in this case, and the plant endophytes. So during seed maturation, that just endophytes move during seed formation. So we had to sample also endophytes at blossom, because we don't know also when this movement of microbes would happen. Is it at flowering or later? And so shoot endophyte at the bloom, harvest, and then we took the seeds, the outcome of the same trees at the end of the season. And we did the same prediction model. And again, this is of course interesting because we have a lot of unknown. We don't know the source of the seed microbiome in this case. But you can see here that we have almost 22% or 22% of the ovary, so the female contribution. Pollen is way too less. And then shoot at harvest. So this is the time really when the fruit is maturing or mature and the seed is still developing, because for apple, seed actually continues to develop inside the fruit way later in the season. And so in this hypothesis, we never published this data, but in our hypothesis that this is really just coming from the mother plant, and even though pollen just gives a little bit more than even the shoot endophytes at blossom, it still contributes, but it's way less compared to what comes from the mother plants, either asexually or sexually. And so to conclude this, we see phylosymbiotic patterns, we see introgression, that's what I just mentioned, we see a conserved microbiome. All of these indicate a sort of co-evolution of plants in general, but of course in this case it's the apple and the microbiome and the Malus species themselves. We can predict with all of this, with phylosymbiosis, with the breeding programs that we also see, that you can predict based on the phylogeny of the plant or the way you, or the breeding lines, you can predict what is the outcome that will come in the future, which would then, this is also a topic then now that is being discussed. Can we breed plants for the microbiome? Can we breed plants with an intention to get a certain set of microorganisms? And I think this is really feasible. And again, the microbial inheritance to explain this process is actually important, so we can understand if the really co-evolution happened between plants and microbiomes. And so this is not that paper that I just discussed, but this microbial inheritance I'm going to talk about tomorrow in Stockholm University. But if you cannot, I hope you can join if you can. But if not, you can read the paper there. And at the end, of course, the acknowledgement, the co-authors of this paper and the funding, obviously, the Marie Curie. And thank you for your attention.